All right. So thank you again, Ilan, for organizing this beautiful event. Thank you, Ronit, for the pointer, and thank you for staying and not going out for coffee. So today I'm going to talk about physics, biology, and multiple sclerosis, and something else, I guess. But and this is obviously in memory of Eshel. Eshel was my colleague and was also my teacher. I mean, he was my professor for thermodynamics at my undergraduate studies. In fact, I wanted to go and do my PhD with Eshel, but he was busy, so I ended up doing something else, something from his former lives of solid state physics, and life meant it to be like that I went into biology as well. And when I'm thinking about the title of this conference and what it means, at least to me, and I think that we're all focusing about the second point about the complexity of how can we understand biology in the complexity of biology with the very simplified physical tools or very simplifying ideas coming from physics. But I think that biology also holds another thing for physicists like myself, trying to go into biophysics or physics of living systems, because we can find emergent physics, new and interesting physics that is coming up from biology or from biological structures that we study. So in many sense, this is very much inspiring me in my research. And today I'm going to tell you about a story that we were dealing with for the past 4 or 5 years. And these are the collaborators that are on this work. It's about multiple sclerosis initiations. And the funny things that I was just reminded by my student, Rona, that led this work, that she came to my lab from Eshel's office, that Eshel introduced her to me and told her that maybe she wants to do her work with me. So I'm very much grateful for Eshel, also for introducing me, for Rona for doing it, and then that led to this work. So everything is connected somehow. So the brain is very, very complex. Like we heard today, you need to have oxygen, you need to have this ADMP, you need to have the immune system, you need to have everything. And for physicists, this is mind-boggling just to think about the complexity of it. So I'm trying to lower down the resolution and think about it as simplified the things that we study in physics. So we have the neurons and the axons. This is the processing units and wires that are going inside our brain. And as any wires that anyone that played with electrical wires knows, that you need to have a good insulators, because those insulators prevents you from having electrical shocks. And in terms of the biology or in what happens in the brain, is actually our wires, our axons of the neurons are surrounded by an insulating tape. It's called the myelin sheets, right? So when things breaks down in the insulation layer, so we know what happens in the physical world. It burns down and the processing unit is damaged. And what happens in biology is something else. And this is where the immune system is not playing so nicely with us. And this is in diseases like multiple sclerosis. It's an autoimmune disease that practically the immune system is acting as the bad person over there and starting to attack and chew up our insulating layer, which causes further damage. Now, the initiation or the repairment of this disease-like myelin sheets is actually not known. There are many different types. So one of them, obviously, was developed in Weizmann of the Copaxone. But what actually starts all of this disease is a great mystery and influences millions of people around the world. So what is this myelin sheets? What's the structure of this myelin sheets? And it's actually made out of mainly membranes, mainly lipid, fat that is surrounding and just spiraling around our axon. And it's composed of two major components. One is the lipids themselves, the phospholipids. This is the insulating tape. And the other component which is very critical, is the myelin basic protein. It's a specific protein which is charged and is acting as a glue to hook up those phospholipid membranes, those insulating tape. Both of them are known to be very critical and very important for the proper function of the myelin sheets. Now, we got into this business because of old studies that were done in the 1980s and following that showed that in diseased state, meaning in multiple sclerosis and in multiple sclerosis examples, there is a minute, really minute changes in the compositions of those building blocks. It's not that you don't have a membrane, you obviously have a membrane, but there is a different stoichiometry of the different lipids that forms the myelin sheets. Really minute. Now this is a very complicated system. You have many different lipids. There are 7 major lipids over there. There are these proteins and so on. So we got into this business embracing the complexity of the myelin sheets and try to see whether we can have some emergence understanding about why it's being damaged. So this is just the stoichiometry of the different lipids and cholesterol, and you can see here the mole percent of each one of them. So in order to follow that as the physicists do, we created what we call a phase diagram, right? We change each of those components slowly, one by one. Well, I didn't do anything, Rona did. And we wanted to see whether that makes any effect on the structure of this membranes. So we made this model system of making multilamellar vesicle. And with this multilamellar vesicles, we can actually monitor what is the structure of the membrane as a result of modifying the stoichiometry of the lipid. Whether this is something of importance, whether it is something that is an aftershock, whether it's something that is none of importance, that was our idea. So how to measure that? So obviously one way to measure that is to go and collaborate with great colleagues like Ishi Talmon and do cryo-TEM. And you can see here the membranes, how it's spiraled around and make these beautiful onions from the lipids. But the techniques that we like best is called small angle X-ray scattering, is that taking a bunch of those liposomes or multilamellar vesicles and shine X-ray into that and looking into very small angles and see the correlations that are coming from, oh, I lost battery, from the structures that you have. So if we identify the structure that is coming from the detector, we can actually find that this is lamellar stacks of membrane, and we can quantify exactly what is the distance between lamellas and so on and so forth. Now, so the first story is what happens when you don't have this glue, this adhesive glue, this myelin basic protein. What happens when you change the stoichiometry as one find in disease state, right? In disease state, you have less of this myelin basic protein. The lipid composition is known to be different. What will be the structural implications of those little differences? So this is the data that came out. The native forms, the healthy myelin composition gives you these lamellar stacks, as I told you before. But this is where the surprise came. When you go to the disease state composition, what you find is that you also have these lamellar peaks, and we know how to quantify them. They're coming in harmonics of linear harmonics. But you can find also indications for what we call an inverted hexagonal phase. I'll get into that. I know it sounds scary, but it basically tells you that there is another structure in your solution. And the structure is rounded and shaped like tubes, and with specific orientation and so on and so forth. So what is this hexagonal phase? And you can see that it's actually coming in the same sample. You can see both. We can also quantify it by electron microscopy, where you can see the lamellar stacks coming either from the healthy or from the diseased state. But in the same sample, you can also see this really beautiful honeycomb hexagonal phase that is present in the same sample. So what is that hexagonal phase, and how is that related to anything? Well, lipids comes in different shapes and forms and sizes. And depending on the size of your head group and the size of your tail, the length of your tail, and how much it is able to fluctuate, it can make different forms. And that's something that we learned from physics, and physicists study that for the past 60 years or so. If your head and tails are pretty much on the same size, they will form sheets. If your head is too big in comparison to your legs and you want to stack them up like Legos, you will end up with a cylinder, right? If it's the other way around, the cylinder will be out inside out, right? So this is the hexagonal, the inverted hexagonal phase, and it's all depending on the size of your lipids, right? So all of that was done for a single lipid, maybe 2 lipids. Nobody did that for 7 different lipids together like we did, but that's what we did. And then we went into the literature and we looked on how this packing parameter, how much the size of the head to the tail is being changed along all of the compositions that we changed. And we can see that everything that we increased pushed us into the hexagonal phase, and everything that we decreased in composition actually was initially in the lamellar packing and went back, and because we decreased it, we pushed it back. So you can see that there is a big balance between the lipid composition that drives you in the end to the different structure that these membranes can form. I'll get back into that. How is that even relevant to multiple sclerosis in a second? Now, the second story that I want to tell you is whether we can study something from biology to new emergent physics, and this is I'll give you just one example for that. So what happens when you add this adhesive protein? So the idea is that those are the results, and I can go over that slowly with you. And what you can see is that this is the scattering data that you have, and you can see that the scattering data is gradually changing as you add up the protein. We can see that the membrane is compacting because of this protein. We can see arrangements of the protein. And I don't want to get into all of the details of what we found over there. I just want to give you one little detail that how we got a new surprise in physics out of this complex biological system. So the idea that 2 entities, one is negatively charged, one is positively charged, will actually condense and together is coming from a very simplified idea that was tested before many, many times. And the idea is that in solution, like we have in our body, everything that is charged is heavily screened by the ions that are in the solution. So if we have a charged membrane and a charged macroion, it can be this myelin basic protein, it could be a DNA, it can be a virus, it can be a different other proteins. What happens in solution when 2 of those are coming together, it's actually entropy that drives them together, because the release of all of those counter ions is actually going to play a major role in the condensation. But this comes with an equation, and this is what is beautiful about it. It's come with the equation that the balance between entropy and electrostatic will cease as the charges coming from, let's say, the protein will match the charges that are coming from the lipids. So that ratio, this charge ratio, should be 1 when the condensation will stop. Now, what happens with our membrane? And with our membrane, we find that this is different. It's not like what happens before with DNA. It's not what happened before with viruses and so on, where the complex membranes with the macroions. Here we see that it sees at 1/2, and after that it proceeds. And that gives us another indication that there is a strong correlation or a strong interaction between those specific proteins, one to themselves and to the membranes, that control the condensation. This is the new emergent physics that is coming out of a very complicated biological system. So the last story that I want to make, and I'll finish by that, is that how is that even related to multiple sclerosis? So the idea here is that this hexagonal phase that I was talking about is actually something that breaks the symmetry of the flat membrane. So the way that we look on the formation of hexagonal phase is actually of building up and opening up the membrane in order to make these tubes. So if you're looking on a flat membrane, it has what we call a zero spontaneous curvature. And when we're looking on an hexagonal phase, it's something that is highly curved. And when it's highly curved, it makes strong roughness to the membrane itself on the way to go to the hexagonal phase. Now, how is that coming to effect? So this is our in vitro measurements of our membranes that we did, and you can see that this in the normal case, normal lipid composition, everything is round and nice and regulated. But when you go to the disease lipid composition, everything becomes to be like a flower, right? Everything is undulating. There is strong undulation. When we collaborated with Ruth Arnon from Weizmann Institute, and she gave us all of those slices from brains of mice, you can see that in the healthy mice, everything is round and regulated of the myelin surrounding the axon. While when you go to the diseased mice, where they induce EAE into that, induce multiple sclerosis into those mice, you can see this flowering-like pathology that is appearing in those membranes. So what we think about now in terms of initiation of multiple sclerosis, or the cause for the immune system to even start to attack, you need to somehow open up the gate, you need to open up the membrane for the immune system to start and attack the membrane, to start and attack and misrecognize this sheet as an enemy. And for that you need to have some kind of a trigger for the membrane to open up, because it's a barrier. So in our idea, when we go back to this image, that you can, if you just type in Google multiple sclerosis, you get one of the hundreds of different schemes like that. Now you might have an idea, we think that we have an idea, how's that becoming that spontaneously, without any exterior contributor, apart from what is the building blocks that makes up those membranes. So it's all coming down to physics in the end. So by that, I just want to thank again my students, particularly Rona and Ram, which is somewhere, another student that contributed to this work, my great collaborators, that none of that could be done without them, and my great students, funding, and you for your attention. Thank you.
Questions? Questions?
They have no more oxygen.
So, what do you know about mutations on these proteins? And basically, people have tried to change the charge of them, or?
Yeah. So people change the charge of the protein itself, and that makes so I didn't tell the whole story about it, but the protein in the healthy forms may like polymerize the protein one into another into the sheets, in between the sheets. When you do mutation on them, this polymerization is not working that well. So you mess around with the structure as well of this adhesive glue. So people have done that before, not so long ago.
Yes.
So how is this an autoimmune disease? It's more a disease of the composition of the myelin sheets.
Well, the disease, the autoimmunity part of it is a great mystery, right? Because somehow the immune system, which is all around there, never attack the myelin beforehand. But when the process starts, when the myelin is being opened up for some reasons to the immune system misrecognize the proteins there as enemies and start to chewing it up. So it's our belief, needs to be tested more probably with mice and stuff like that, that our logic is actually working in the right direction. But if it is, then it will be very it could serve as a good breakthrough to understand the disease etiology or initiation.
Other questions? Yes.
It's not a question, it's support to what you are proposing. It's known that even if you inject immune cells into the CSF, even autoimmune T-cells, they will circulate and will not do any damage. They will not even enter the parenchyma unless you have it triggering inside the cell.
And the big question is, what is this triggering?
It's chemokine, but you need to signal it, otherwise it will not enter.
Yeah, but what I'm saying is that what we are trying to understand is what initiating it in terms of the myelin itself, that is otherwise a good protective sheet, and whether it can be something that initiated from within the synthesis of the myelin and so on. So we think that since this is done in vitro without any complexity to it, and it still forms this, it's just open up. It's just undulating and showing the same pathology as one could see in diseased animal and diseased brains.
Okay, well, thank you, and thank all the speakers. First of all,